Hello friends, now talk about eukaryotic cells. Okay, eukaryotic include all the protists and plants, animals and fungi. In eukaryotic cells, there is an extensive compartmentalization and of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Okay. Plants and animal cells are different as the former possesses cell walls, plastid, and large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plants. Already मैंने एक लेक्चर में बताया था. Now talk about cell membrane. The detail of the structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, the chemical studies on the scale membrane, especially in human red RBCs, embedded in scientists on the deduce the possible structure of the plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. Okay, main point. The major lipids of our phospholipids are arranged is a bilayer. Okay. Also the lipids are arranged within the membrane with the polar head towards the outside and the hydrophobic tails toward the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition of the phospholipid membrane also contains cholesterol. The lipid compartment of the membrane mainly consists of phosphoglycerides. Later, biochemical investigation clearly revealed that cell membranes also possesses proteins and carbohydrate. The ratio of the protein and the lipid varies constantly and different in human beings. Cell membrane in erythrocyte explain 52% of protein and 40% of lipid. Depending on the ease of extraction, membrane proteins can be classified into integral and peripheral. Peripheral proteins lie on the surface on the membrane, which is integral proteins partially or totally buried in. This is a fluid model plasma. This is a cholesterol. This is sugar, peripheral protein. This is a lipid bilayer, phospholipid bilayer, integral protein. Okay, the membrane. An improved model of the structure cell membrane was proposed by Singer and Nicholson 1972. Year must widely accepted the fluid mosaic model. According to this, the quasi-fluid nature of the lipid embeds bilateral movement proteins within the overall bilayer. This ability to move within the membrane is measured in fluidity. Measured by fluidity. The fluid nature of the membrane is also important from the point of the view function like cell growth, formation of intercellular junction, secretion, endocytosis, cell divisions, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Neutral solute may move across the membrane by the process of simple and diffuse along the concentration gradient. From higher concentration to lower, this is diffusion. Water may also move across the membrane from higher to lower concentration. This movement is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer and they require carrier protein of the membrane, this is a facilitated their transport across the membrane. A few ions or molecules are transported across the membrane against the lower to higher concentration gradient from the lower to higher concentration. This is called the active transport. Example is sodium potassium pump. Now talk about cell wall. A non-living rigid cell structure called the cell wall forms an outer covering of the plasma membrane of fungi and plants. Cell wall not only gives shape to cell, protects from the cell mechanical damage and infection. It also helps cell-to-cell interaction and provides barrier to undesirable macromolecules. Algae have cell wall made up of cellulose, galactans, minerals like calcium carbonate, while the other hand consists cellulose, hemicellulose, pectins, proteins. The cell wall of young cell plant primarily is capable of growth, okay, which gradually diminish as the cell mature, and secondary wall is formed in the inner towards side of the cell. The middle lamella is the layer mainly calcium pectate which holds up glue the different neighboring cells together. The cell wall of the middle lamella may be transport of the plasmodesmata, which is connect cytoplasm of neighboring cells. Okay, now talk about endomembrane system. While each of the membranous organelles is distinct in its structure and function, many of these are considered together is an endomembrane system because their function is coordinated. The endomembrane system include an ER, Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, peroxisome are not coordinated with the above compartments, these are not considered part of the endomembrane system. Okay. Electron microscope study of eukaryotic cells reveal the presence of the network of the reticulum of the tiny tubular structure scattered in the cytoplasm. This is called the endoplasmic reticulum or ER. ER divides in the intercellular space into two distinct compartments: luminal inside ER, extraluminal cytoplasmic compartment. The ER often shows the ribosome attached to the outer surface. The endo-ER bearing ribosome on the surface called the RER, rough endoplasmic reticulum. In the absence of ribosome, it appears smooth and called ACR. RER frequently observed in the cells actively involve the protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The ACR is the major site of the synthesis of lipid. In animal cells, lipid like steroid hormone and synthesis ACR. Now talk about the endoplasmic structure. This is the nucleus, this is the nucleus pore, this is the RER because this is a ribosome is attached, this is a ribosome, and this is because here ribosome is not present, that's why this is ACR. Now talk about Golgi apparatus. Camillo Golgi in 1898 first observed the densely stained reticular structure near the nucleus. These were later named the Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of the 0.5 micrometer to 1 micrometer diameter. These are scattered parallel to each other. A varied number of cisternae are present in the Golgi complex. The Golgi cisternae is constantly arranged near the nucleus in a distinct convexes or the forming. This is a cisternae. Okay. Golgi apparatus. This is the structure of Golgi apparatus. The forming phase and the concave trans or the maturing phase sees the trans phases or the organelles are entirely different but interconnected. The Golgi apparatus principally perform the function is a packaging materials to be delivered either at the intercellular targets or secreted outside the cell. Materials to be packaged in the form of the vesicle of the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in the close association with the ER. A number of proteins synthesized by ribosomes on the ER are modified in the cisternae of the Golgi apparatus before the release of the trans phase. Golgi apparatus is the most or important site of formation of the glycoproteins and glycolipids. Important point. Now talk about lysosomes. Okay, this is the membrane-bound vesicular structure formed by the process of packaging and in the Golgi apparatus. The isolated lysosomal vesicles have been found to be rich in the almost all types of hydrolytic enzymes, hydrolyze, lipase, protease, carbohydrates, optimally active in the acidic pH 4 to 5. The pH is 4 to 5. These enzymes are capable to digest carbohydrate, proteins, lipids, and the nucleic acid, nucleus. Now talk about vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excreted product, and other material in the useful for the cell. The vacuole is bound to a single membrane called tonoplast. Important. What is tonoplast? In plants, the vacuoles can occupy the up to 90% of the volume of the cell. Main point. In plants, tonoplast facilitated the transport of a number of ions and the materials against the concentration gradient into the vacuole. Hence, the concentration of the significantly higher in the vacuole than in the cytoplasm. In amoeba, main note point. In amoeba, contractile vacuole is important for excretion in many cells. Sport is food vacuoles formed in the engulfing and the food particles. Thank you. Okay. Next day, mitochondria start. Mitochondria, plastid, cytoskeleton, cilia, flagella. Okay. Thank you.